Uh, Hello, welcome everyone to my channel. Today is the third lecture of erythropoiesis. Now we are going to see stages of erythropoiesis. First stage of erythropoiesis is pro-erythroblast or pro-normoblast. Pro-erythroblast is the first cell derived from CFUE. And what is CFUE? CFUE is committed pluripotent homeopathic cells, and it's from them it divided into colony forming blastocysts, and the colony forming blast unit E will produce RBC. So CFU is colony forming unit E. So the first cell derived from colony forming unit E, it is a large cell. It is very large cell with a diameter of 20 micron. Now diameter of 20 micron is, and the mature RBC diameter is almost like 7.2 micron. So it is way too larger than the previous the mature RBC. Now its nucleus is large. It has nucleus pro-erythronormo pro-erythro or pro-blast or pro-normoblast. It also have large nucleus. Now mature RBC doesn't have large nucleus and occupy the cell almost completely. It has such a large nucleus that it occupies the cell completely, like its nucleus is as large as itself. And nucleus has two or more nucleoli and reticular network. Now it also have nucleoli is present in nucleus, as we already know. And reticular network means reticular is endoplasmic reticulums. And pro-erythroblast does not contain hemoglobin. Now the CFUE first cell, CFUE, when it gives the first cell, it is pro-erythroblast. This is a nucleus of pro-erythroblast which have two nucleoli. It can have more nuclei, and it also doesn't have hemoglobin. Hemoglobin production is not started in this stage of erythropoiesis. Cytoplasm is basophilic. Cytoplasm is basophilic in nature. Cytoplasm is basophilic. Basophilic means it will take basic strands. So it is called basophilic. Basophilic means base loving in nature. Pro-erythroblast multiplies several times and finally form the cell of the next stage. These pro-erythroblasts, they multiply several times. That means they divide so many times and form the next stage, early normoblast. Now pro-erythroblast or pro-normoblast, this will give early normoblast. Pro-erythroblast, early normoblast. Synthesis of hemoglobin start in this stage. Now synthesis of hemoglobin is getting started. However, appearance of hemoglobin occur only in intermediate normoblast. In early normoblast stage, the synthesis of hemoglobin is started, but it doesn't appear. It only appears in intermediate normoblast stage. Now early normoblast become smaller with diameter of 15 micron. Now the diameter of pronormoblast was 20 micron. Now it come in next stage, it make early normoblast, and the diameter of early normoblast is lesser than the pronormoblast. It is 15 micron. Nucleoli in nucleus disappear. Now as we see in erythrocytes or pronormoblast, there are two nucleoli in nucleus, but in this stage there is the nucleoli disappear. No nucleoli present in this stage. The chromatin network get condensed and become dense. The chromatin network, the network present inside the nucleus, the chromatins, they get condensed and become dense. They become dense. Dense means they appear darker. Cytoplasm is basophilic in nature. Even in this stage also, cytoplasm basophilic. So this cell is called basophilic erythroblast. This cell in this stage cell also called as basophilic erythroblast. This cell develop into intermediate normoblast. Now basophilic erythroblast will develop next stage as intermediate normoblast. Now the third stage, intermediate normoblast. The cell is still smaller with a diameter of 10 to 12 micron. Now the cell first it started with 20 micron, then in next stage it got 15 micron, and in third stage it became 10 to 12 micron. The cell is getting smaller. Nucleus is still present. Nucleus first in first two stages nucleus was also present, but in second stage nucleoli disappear. In this stage also nucleus is present, but the chromatin reticulum show further condensation and hemoglobin start appearing. Now the production of hemoglobin was already started beforehand, but hemoglobin start appearing only in intermediate normoblast stage, and the chromatin further condense. They are condensing. Cytoplasm is already basophilic. Now because of presence of hemoglobin, it is stained with both acidic as well as basic stain. So this cell is called polychromophilic. So see, first first two stages, this is basophilic. Even second stage also the cell is taking only basic strength, so it's basophilic. But in third stage, as hemoglobin start appearing, hemoglobin already present and it start appearing, so it will take acidic strain also. So taking both acidic and basic strain, it's called polychromophilic. See, first let me give a little bit this one. First, CFUE, that means colony forming unit E. It will further divided into proerythrocytes. Further it converted early normoblast, then intermediate normoblast. Next stage we will see polychromatic erythroblast. See, polychromatic erythroblast, which means it will take both acidic and basic strain. This cell is developed into late normoblast. Now this intermediate normoblast will develop into late normoblast. Now late normoblast, diameter of this cell decrease further about 8 to 10 micron. Nucleus become very small with very much condensed chromatin network, and it is called as ink spot nucleus. Now see, in this stage nucleus, in first stage nucleus was very large and it was covering the almost whole space of the cell. But as we started going down, as we started going in further divide next stages, so nucleus, as cell is also decreasing, size of nucleus is also decreasing, and in this stage late normoblast, it became a very small, and the chromatin, they become very condensed. They are no longer reticular, they are very condensed chromatin network, and it is called as ink spot nucleus. Ink spot means little spot. Now quantity of hemoglobin increased and cytoplasm became almost acidophilic. Now as hemoglobin quantity increases, first cell was basophilic, but as appearance of hemoglobin in intermediate stages, it became both acidic and basophilic, acidophilic or basophilic, but which is also called polychromatic But in this stage, it's due to appearance of too much hemoglobin, the cell almost became acidophilic. So now the cell is called orthochromatic erythroblast. In third stage it was called polychromic erythroblast, but in fourth stage it will called as orthochromic erythroblast. In final phase of late normoblast, just before it passed to the next stage, nucleus disintegrate and disappear. In final stage, first nucleus was very small, but in final stage, at is about to enter in reticulocytes, the nucleus became disintegrate and it disappeared. Process by which nucleus disappear is called pycnosis. By the process of pycnosis, nucleus disappear. The final remnant is excluded from the cell. The final remnant is excluded from the cell, which means the final remnant which are present, the portions, the parts of the cells which are present, organelles or other things which are present in the cell, they are excluded. They are taken out of the cell and digested by macrophages, and late normoblast develop into reticulocyte. In late normoblast, it will go to the next stage, reticulocyte. Reticulocytes is otherwise known as immature RBC. These are called as immature RBC. It is slightly larger than the mature RBC. So it is slightly larger than. First cell was too much larger than the RBC, but coming to the fifth stage, it is slightly larger than the immature RBC. Cytoplasm contains the reticular network or reticulum. In cytoplasm, there is a reticular network which is formed by remnant of disintegrated organelles. Those organelles which are disintegrated, their remnant make the cytoplasm appear as a reticular network or reticulum, and due to reticular network, it is called as reticulocyte. This stage is called as reticulocyte due to appearance due to reticular network, and reticulum of reticulocyte stained with supervital stain. Supervital stain means those vital stains are those stains which work on living without killing the cell. In newborn babies, reticulocyte count is 2 to 6% of RBC. The count decreases during the first week after birth. Later, so in newborn babies, as they are born, the reticulocyte count is 2 to 6%. The count decreases during the first week after birth. Later, the reticulocyte count remain constant at below 1% of RBC. So after they born in the first week, there so many changes are going on in the body of the, and reticulocyte count, it decreases to 1%, and after that it remain constant below 1%. It remain constant. Number increase whenever production and release of RBC increase. So whenever the RBC production increases, its number also increases, because definitely, because it's a immature RBC. So after this stage, mature RBC is going to form. To forming mature RBC, we have to increase the number of immature RBC. Reticulocyte is basophilic, and again this is basophilic due to presence of remnant of disintegrated Golgi apparatus, mitochondria and other organelle of the cytoplasm. So reticulocyte basophilic due to presence of, ये क्यों है बेसोफिलिक? फर्स्ट तो एसिडोफिलिक, ये हीमोग्लोबिन की प्रेजेंस की वजह से लेट नॉर्मोब्लास्ट हो गया था, ऑलमोस्ट एसिडोफिलिक हो गया था, बट रेटिकुलोसाइट बेसोफिलिक फिर से क्यों हो गया? क्योंकि जो भी डिसइंटीग्रेटेड पार्ट हैं गोल्जी एपरेटस के, माइटोकॉन्ड्रिया के, अदर ऑर्गेनल जो भी साइटोप्लाज्म में होते हैं, कि उनकी वजह से वो जो ड्यूरिंग दिस स्टेज उनकी वजह से ये बेसोफिलिक हो जाता है. ड्यूरिंग दिस स्टेज cells enter the blood capillary through capillary membrane from the site of production by diapetesis. So इस स्टेज में क्या होता है, जो सेल्स होती है, जो आरबीसी प्रोडक्शन के लिए सेल्स होती है, वो ब्लड कैपिलरीज में कैपिलरी मेम्ब्रेन के थ्रू जाती हैं थ्रू डायापिडेसिस. डायापिडेसिस मीन्स अमीबॉइड मूवमेंट की जैसे अमीबॉइड मूवमेंट करके वो कैपिलरी ट्यूब में कैपिलरी मेम्ब्रेन के थ्रू जाती हैं. नेक्स्ट इज mature erythrocyte. Mature erythrocyte, the reticular network disappear and the cell become mature RBC and attain the biconcave shape. Now, reticular network, those remnants of Golgi apparatus or reticular sites, these all disappear and the cell become mature and attain a biconcave shape. Biconcave shape means this is like a concavity is present from both sides in the center. Diameter of cell is decreased 7.2 from 22 it is come to 7.2 micron. A mature RBC is with hemoglobin but without nucleus. Now nucleus is completely disintegrated. RBC is filled with hemoglobin. It requires 7 days for development and maturation of RBC from erythroblast. So from first stage to mature RBC, it takes 7 days of development and it requires 5 days up to the stage of reticulum. Reticulocyte take 2 days to become mature RBC. So 5 days for other stages and 2 days only for reticulocyte to become mature RBC. First, if we see this, we will able to understand all. First stage of erythrocytes. So pro-erythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte and mature RBC. Diameter. In first stage it was 20 micron. Then in next stage it became 15. In third stage it became 10 to 12 micron. In fourth stage it became 8 to 10 micron. In fifth stage it became 7 to 7.5 micron. And in last stage it became 7.2 micron. Now nucleus and staining property. In first stage, which is pro-erythroblast, nucleus has 2 or more nucleoli and chromatin network. But in second stage, nucleoli disappear, dense chromatin network is present. In third stage, further condensation of chromatin of network, and in fourth stage, small with very condensed chromatin ink spot nucleus is present, and from reticulocyte nucleus get absent. From in reticulocyte and mature RBC nucleus is absent. Next, staining properties. So in pro-erythroblast it is basophilic. Second stage also basophilic. Third stage is both acidophilic and basophilic. We call this chromophilic. Then later normoblast is acidophilic. Reticulocyte is basophilic, and then mature RBC is also acidophilic. This is very important. Now because of absence of all the reticular structures and only hemoglobin is present, that's why it became again acidophilic. Important is event in first stage, synthesis of hemoglobin started in first stage, like in pro-erythroblast. Sorry if I said wrong before, but in pro-erythroblast itself, synthesis of hemoglobin started. In second stage, nucleoli disappear. In third stage, hemoglobin start appearing. It starts synthesizing in first stage, but it start appearing in third stage. Next, reticulum is formed cell intercapillary from the site of production through diapetesis. So, reticulum, due to destruction of Golgi apparatus or ER, these reticular structures are formed in cell, and in this stage cell enter the capillary from the site of production through diapetesis and in last is the reticulum disappear and cell attain biconcavity. Next, in next class we will study factors necessary for erythropoiesis.